Hello and welcome to This Week in Science, a YouTube series where I talk about different scientific papers that are less than a month old, and we'll talk about some different breakthroughs and kind of revolutionary papers that were published recently. So this is the episode for the week of January 26, 2020, or the last week of January in 2020. So I have 5 different interesting articles for you guys today. There's one about extraterrestrial ribose, there's arthritis treatments, exercise, and a protein that was found to be associated with exercise. There's also one about platypus distribution and another one about the effects of heat and pests on plants. So let's go ahead and begin, and I'll share my screen with you guys so you can go ahead and follow along and see the different papers that I have picked out for this week. So first we have a research article titled Extraterrestrial Ribose and Other Sugars in Primitive Meteorites. This was authored by Furukawa et al., and this is in the Proceedings of the National Academy of Sciences. So let's go ahead and start to talk about this paper. So basically the main discovery here is that authors found ribose in meteorite, and previously substances and molecules such as like amino acids, nucleotides, and glucose have been found in meteorites, but this is the first evidence of ribose in meteorites. And this is significant because ribose is a sugar that's needed to form RNA. And if you recall, DNA turns into RNA, or DNA codes for RNA, which codes for proteins, basically. So it's an important part of our gene expression sort of pathway, if you will. So this is significant again because this is the first time that they've found evidence of RNA from an origin other than obviously on Earth. So I'll go ahead and show you some of the figures that they have here. You can see the different structures of the actual sugars here. And then this is, and I should point out, they found other sugars too, but obviously ribose is going to be the one of interest. That's the one we're discussing. So you can see here they did gas chromatography and mass spectrometry to find, to characterize the different molecules that they found in this meteorite. And you can see they have reference spectrums and standards, and then they have 2 different meteorites that they've looked at. So basically what this is showing is that you can match the experimental spectrum, the experimental output, to a standard, and from there you find, okay, there's ribose in this meteorite. And then later on, the authors, it's discussed in this area, they also used isotopic analysis to basically confirm that this ribose was actually extraterrestrial in origin, that it's not from Earth. They go really in depth on this, so if that's something that you're interested in, definitely read the paper. But this article I found interesting because further research in this area might help clarify how life kind of came to be on Earth. I'm sure you guys are familiar with a set of experiments that kind of showed that you could make essential compounds just from certain changes in temperature and water and evaporation and electricity, kind of trying to simulate the atmosphere that Earth used to have when it was, just formed. So I think that's something that's very interesting, learning more about the origin of life on Earth. I'm also interested to see if there's going to be any reviews of this study along with other ones that kind of maybe put together the pieces a bit more and see how it fits in with what we already know, and if it either supports existing theses or if it actually kind of takes away from them and has a sort of counterpoint in here. So that will be definitely interesting to see. I'm looking forward to those reviews. So let's go ahead on the next one. Here we have alpha KLOTHO and STGF beta R2. Treatment counteract the osteoarthritic phenotype developed in a rat model. This is published in Springer, and this is by Martinez Redondo et al. So, the authors start out by mentioning that currently arthritis really has no cure. It's more so treated symptomatically. So they looked at these two different molecules. They have alpha KLTHO, which prevents degradation of the extracellular matrix, which is the sort of area surrounding the cells. It can have a structural function. It can have an impact on cell signaling. It has various molecules in it, so on and so forth. And they also looked at STGF beta R2, which encourages cell proliferation and growth of cartilage cells. So that was a very interesting approach to me. Is they take one molecule, the alpha KLTHO, which prevents damage and degradation and death, and then they take another molecule, the STGF beta R2, which encourages growth. So they're both kind of having a defensive and offensive approach at the same time, if you will. So what they did is they injected rats with a viral vector carrying the information to encode for these molecules, the DNA for these molecules. And there is a lot of controversy about the safety of viral vectors, but the main reason that they do use viral vectors is because they're very good at integrating whatever DNA the virus is carrying into the cells. In terms of safety, insertional mutagenesis is really the main concern, but that's still something that's being kind of worked out in the gene therapy academic circle, basically. But there are quite a few clinical studies for viral vectors, so there is at least some degree of safety where the government is okay with having us run studies on those, basically. So they used methods like Western blot and immunohistology to analyze the treatment, basically, and they showed that the treated rats had more new cells forming and also thicker cartilage when compared to control rats. So the KT are the treated rats and the sham are the non-treated rats. So you can see here, I believe they use PCR to kind of characterize gene expression. They also used, obviously, lots of staining and immunohistochemistry, and then the Western blot. But you can see here, let's take this for example, the Ki67 is basically a marker for newly forming cells, proliferating cells, and it shows up where the nucleus is relatively. So you can see here there's just a lot of noise. I assume this is skin. Skin, obviously, you're always having new cells forming because you're constantly shedding your outer layer of skin. But you can see here a little bit deeper into the skin, there's actually these newly forming cells, and you can make them out individually. So that's very interesting. They can support that they have actual new cells forming, and then they have, this looks like it might be trichrome staining. So you can see that the in the sham, there's really not a whole lot of cartilage, whereas here you can see that there's more. I think that they yes, they quantified it here, and it is a significant difference when compared to the sham. So that's Obviously good news. So the authors also discuss later on in the paper how they have started looking at human cells in culture, so in vitro, and so far that they say that it looks promising, but they didn't really have a whole lot of data for that, so hopefully they continue these studies and they're able to either do more in cell culture or even move on and do a clinical study. So next up we have a article called Sustrins are Evolutionary Conserved Mediators of Exercise Benefits. This is by Kim et al. In Nature Communications. This is a very interesting article. I think this was my favorite one so far that I've looked at this week. So these researchers basically found a protein that can mimic the molecular and physiological aspects of exercise, and these are known as sestrin. They're actually a family of proteins, so they're sestrins, I should say. So as we know, exercise has lots of both physical and physiological as well as psychological health benefits. So I think this is something that could really be used for like elderly people and people who are injured or disabled and cannot exercise. Hopefully they can still get the benefits of exercise, because without that, your muscles start to weaken and you're subject to muscle wasting and so on and so forth. So I think that's something that would be really interesting to see if we could potentially use this therapeutically in the future. So they tested this on 2 different animal models. First they used Drosophila or fruit flies, and then later on they used mice. So with the with the Drosophila is what they started with. They basically, let me pull this picture up larger. What they did is they looked at the run span, which is maybe a bit misleading. It's the amount of time that they can fly around before they get fatigued and land, basically. So the flies were stimulated to make them fly, sort of as a training for exercise. And those that didn't have sestrin did not increase their flight times, unlike the ones that did have sestrin. So they basically did a knockout here and prevented the animal from making this protein. So this control, UN means that they did not get any exercise. Control exercise means that they were able to exercise previous to this experiment. So the ones that have been exercising for 25 days. And then these are without sestrin, both ones that didn't have the chance to exercise and ones that did. This was found to be a insignificant, there was no significant difference between those 2 groups. However, there was a significant difference between the control that were able to exercise and the control that wasn't. And you can see here that the values are even smaller for the sestrin animals, so we can assume that there's also a significant difference there. You can also see they did Western blots, and they looked at things like performance as well. So I thought that experiment alone was very interesting. However, they also went ahead and they looked at mice as well. So let's pull up the figure for the mice. This is one. So they did one with voluntary running and one with involuntary running. I'll just show you the voluntary ones here. But if you are interested in the other aspect, you can go ahead and read this paper yourself, of course. So in mice, they found that sestrin was actually required to, again, obtain certain exercise benefits. So they didn't just look at performance, but they also looked at things like insulin sensitivity, which is associated with exercise due to previous research. And they also looked at exercise performance, respiratory efficiency, so on and so forth. So you can see here they also did this SN1KO is a sestrin knockout. So again, basically preventing the animal from making a certain sestrin protein. So here you can see they looked at both speed, distance, and time. So all of these are different aspects of running, basically different ways to measure them running. So for all of these groups at week 1, there was no significant difference. Week 2, there started to be significant differences in terms of speed and distance, but they still had no significant difference in terms of time. And it looks like it may be due to this kind of outlier data point, I'm not sure, but that would be interesting to see. But regardless, when including all the animals, which is what you need to really do with data, there's no significant difference. However, by week 3, each category did have a significant difference. So that was interesting to me that, of course, they all start out the same because they're basically training these mice to run longer and faster and for a longer distance. So I thought that was really interesting. They also did show improved insulin sensitivity and respiratory efficiency as well. You can see over here. And then they also, let me go back. So that was voluntary, then this is the forced running. But the authors also do discuss quite a bit about the actual pathway that cestrin is involved in. So if that, again, is something that you're interested in, go ahead and read the paper. So next up, we have a stitch in time, synergistic impacts on platypus metapopulation extinction risk. This is published in Biological Conservation by Bino et al. So as the authors start out, the platypus is listed as near threatened under the IUCN Red List, and they basically go on to say that not a whole lot is known about platypus distribution, but there have been recent declines and areas where there's no longer sightings that have kind of started to cause concern. They hypothesize that the cause of the declines is linked to things like fragmented habitats, so things like putting dams in rivers, also fishing, drought, and invasive predators such as certain types of foxes and dogs. So they go on to discuss that declines are expected to be worse in areas with drought issues, and that can lead to local extinction. And let me pull up this figure here. They show the years of last sighting, and you can see there's some where it's possible there's been a local extinction. And this is a percentage of if the platypus is extinct or not, basically. So the red areas are 70 to 100% extinct. So that's based on, again, previous data of sightings and whatnot. So, yes, they continue to go on and talk about how they surveyed the platypus and what areas they went in, and talk about their methods, of course. And basically what they end up with is that currently the platypus is considered near threatened by the IUCN Red List, but they urge considering a change to vulnerable, and they go over the specific criteria for the IUCN Red List because there are quite a few. Some of the resolutions that they propose are things like, obviously we need to, take into account urbanization and climate change, because those are really strongly acting on the platypus. We also have other concerns such as invasive species, and the researchers also discussed that there's not a lot of research about platypus reproduction, so additional research could be beneficial to having a more accurate idea of the actual threat to the platypus, as well as ways that we could potentially start conservation efforts, because at one point they do mention that it's better to act preemptively and keep these platypus, fairly protected before going into a near extinction crisis and then trying to save them. It's easier to start now. It's basically the conclusion that they have. So that's really it for that paper. And then next we have insect herbivory antagonizes leaf cooling responses to elevated temperature in tomato. This was published in Proceedings of the National Academy of Sciences by Havko et al. So basically, the researchers wanted to look at how plants defend against bugs during warmer temperatures, so things that would be relevant in a sort of climate change scenario. We know, researchers basically know how plants cool themselves off and how they defend against bugs, but not at the same time. So that's something that these researchers wanted to look at, because that's going to be very important for climate change models. And if the experiment doesn't turn out very well, we might want to consider looking at what solutions we might have, basically. So in terms of defense from bugs, plants typically release a hormone called jasmonate. That's how they defend themselves. And to defend themselves against high temperatures, plants will raise their leaves, trying to get away from the hot soil, if you will, and then they'll also open their stomata and allow evaporation, kind of like how humans sweat. So those are the ways that previous research has substantiated how plants defend against different conditions. So in this research, specifically this new paper, the researchers used tomato plants. Those are important agriculture. They're quite a widely grown crop, so that was a good sort of pick to kind of simulate what would happen. They used hornworms as the pest in this case, and they were used on both control plants and then plants that were at an elevated temperature. So the WT is wild type, and then this is a different type of plant, but this is the control temperature, and then this is elevated temperature. So they had a control plant that was just going to be placed at elevated temperature. They had a plant that would have hornworms on it, and then a plant that would be wounded and not have any pests on it anymore currently, basically. So the elevated temperature that they chose here was 100 degrees Fahrenheit. The plants did release jasmonate, but they didn't attempt to cool themselves off, really. They didn't open their stomata or lift their leaves very much. So that was something that the researchers found as interesting. The plants don't try to cool themselves off when they are under attack by bugs in this specific condition, basically. So they found that the plants still do photosynthesize, but the crop productivity still decreases as well. So they said that they were interested in looking into that a bit more as a further sort of follow-up project as well. You can see they use different sort of characterization methods, such as Western blot. But yeah, that was really the main figure, was the one that I showed you earlier. So that is all we have for this week, and I will post another video for you guys next week. Thank you, and goodbye.